Hello and welcome to some biology class. Friends, today we shall discuss about photophosphorylation. The ATP required for carbon reduction and other metabolic activities of chloroplast is synthesized by photophosphorylation. Photophosphorylation is the light-dependent synthesis of ATP by chloroplast. In addition to using ATP along with NADPH for reduction of carbon dioxide, a continuous source of ATP is required to support other metabolic activities in chloroplast. In chloroplast, electrons are continuously supplied from water and are withdrawn as NADPH. This flow-through form of electron transport from water to NADP plus through photosystem 2, plastoquinone, cytochrome B6F complex, plastocyanin and photosystem 1 is known as linear or non-cyclic electron transport. Non-cyclic electron transport is associated with photolysis of water, release of molecular oxygen and reduction of NADP plus to NADPH. Synthesis of ATP in association with non-cyclic electron transport is known as non-cyclic photophosphorylation. In photophosphorylation, ATP is synthesized from ADP and inorganic phosphate with the help of enzyme ATP synthase, which is also present in the thylakoid membrane. Photosystems 1 and 2 in the thylakoid membrane are not physically linked as implied by the jet scheme, but they are segregated into different regions of the thylakoid membrane. We know that photosystems 1 are almost exclusively present in the stroma lamella. Some of them are also present at the edges of grana lamella, whereas photosystems 2 are predominantly present in the appressed regions of the grana lamella. A consequence of this heterogeneous distribution of photosystems in the thylakoid membrane is that photosystem 1 can transport electrons independently of photosystem 2 in a process called as cyclic electron transport. Cyclic electron transport begins with the photoexcitation of P700. P700 is the reaction center chlorophyll of photosystem 1. Excited P700 is rapidly oxidized by iron sulfur protein called ferredoxin. From ferredoxin, electrons are transferred to a recently discovered protein called PGR5. From PGR5, electrons are transferred to plastoquinone and from plastoquinone to cytochrome B6F complex. From cytochrome B6F complex, electrons are cycled back to photosystem 1 through plastocyanin. This type of electron transport is known as cyclic electron transport. As these electrons passes through plastoquinone and cytochrome B6F complex, cyclic electron transport also contribute to the proton gradient, which drive the synthesis of ATP. Synthesis of ATP in association with cyclic electron transport is known as cyclic photophosphorylation. It is thought that cyclic photophosphorylation is the source of ATP over and above that required in the carbon reduction cycle. Photophosphorylation was discovered by Daniel Arnon and his co-workers in 1950s. This figure shows the organization of the photosynthetic electron transport system in the thylakoid membrane. This is photosystem 2. PQ for plastoquinone. This is cytochrome B6F complex. PC stands for plastocyanin. Photosystem 1 and ATP synthase, which is also known as CFOCF1. That side it is stroma and this side it is thylakoid lumen. During photosynthetic electron transport, there is accumulation of protons in the thylakoid lumen. So the lumen become more acidic, whereas the stroma become more alkaline. This difference in the concentration of protons constitute the proton gradient. The resulting proton gradient is then used to drive the synthesis of ATP in accordance with Mitchell's chemiosmotic hypothesis. There are two principal mechanisms that accounts for the accumulation of protons in the thylakoid lumen. One is the oxidation of water. For each molecule of water oxidized, two protons are deposited in the lumen. The second one is plastoquinone cytochrome proton pump. The precise mechanism by which protons are moved across the thylakoid membrane by cytochrome complex is not well understood. Several models have been proposed to explain the movement of protons across the thylakoid membrane. The widely accepted model is known as Q cycle. If protons move through Q cycle, then for each pair of electron passing from plastoquinone through Reus iron sulfur center and cytochrome to plastocyanin, 4 protons will be translocated from stroma into the thylakoid lumen. If the scheme is correct, then the transfer of each pair of electron by this non-cyclic process from water to NADP plus will contribute 6 protons to the gradient. 4 protons are derived from Q cycle and remaining 2 from oxidation of water. In case of cyclic electron transport, as there is no oxidation of water, so the transfer of each pair of electron by cyclic process will contribute only 4 protons to the gradient. It is generally agreed that 3 protons must be transported through ATP synthase in order to drive the synthesis of each molecule of ATP. Then, for each pair of electron passing through this non-cyclic process, 2 molecules of ATP will be synthesized. The precise stoichiometry is, however, difficult to determine because of the uncertainty in the relative proportion of cyclic and non-cyclic electron transport at a specific moment. According to Peter Mitchell's chemiosmotic hypothesis, the total energy available for ATP synthesis is called proton motive force. Proton motive force is the sum of the transmembrane electrical potential and the proton gradient. Most of the evidences suggest that the steady-state electrical potential in chloroplast is relatively small. So, most of the proton motive force is derived from the proton gradient. So friends, this is all about photophosphorylation. For more updates, please subscribe my YouTube channel, Sun Biology Class.